This is chapter 1. In this chapter, we're going to be looking at the nature of science and also looking at the definition of biology. The first question we're going to look at is, what is biology? In biology, this term we can separate into two parts. The last part of biology, the ology part, stands for the study or science. The first part of this term, bio, stands for life. So if we break this term down and look at the definition of it, biology is the study of life. We're going to look at these two pieces. First off, we're going to start out looking at science, and then we will look at the life part of it. So what is science? Science is a body of knowledge or data that has been collected about the natural world. Important things about this body of knowledge is that it has to be detected, observed, and measured. And this can be done in multiple different ways. In the top image, we have scientists in a lab collecting data on bacteria. We can also collect data in the environment. So we have an ecologist shown in the bottom picture measuring the head length of a bird. So anything that we can measure or detect will add to this body of knowledge. Science is also an evidence-based process for acquiring that knowledge. And this process that we're going to be looking at is called the scientific method. In addition, science is also subject to independent validation or peer review. And we'll talk quite a bit about this peer review. We'll look at it in chapter 1. We will also look at it in chapter 2. The conclusions of science are based on the data that is collected or that evidence. And this data or evidence that is collected is always open to challenge. So other scientists can come in and redo an experiment or do an additional experiment to collect more data, to collect more evidence, to challenge any conclusions that are made by any researcher in any area of science. So because of this, science is a self-correcting enterprise. And this is really important because science is all about gaining knowledge about our world. And we want to make sure that the knowledge that we're adding to our body of knowledge is true, that it makes sense, and that it is updated. So with this, even though science can look at things, it can do research, it can add to this body of knowledge, there are some limits of science. Science can only be applied to observable and measurable data that we collect from the natural world. We cannot use it to look at things related to faith, for example. Science cannot tell us what to do with the information that it uncovers. This goes into ethics or morals. So it can provide no help as to what is morally or ethically right or wrong. An example of this is looking at the human genome. We have sequenced the entire human genome, so we know which genes code for which proteins that give us certain characteristics. Because we have all this information, we can actually design people. So if you would like to have a baby that has blue eyes and blonde hair, we know exactly which genes will lead to those traits. So even though we can do this, then we get into this question of, is this ethically right or wrong? And science cannot tell us or answer those types of questions. So with that, science is a very powerful tool. It allows us to add to the body of knowledge about our natural world, but it does have limitations just due to how it is set up and the nature of science. So scientists do science using the scientific method. And this is a step-by-step process. I'll show you a diagram right here, and this is directly out of the book. We'll go through each of these steps. I'll explain them, add in a few more terms, and then I will give you an example. The first step in the scientific method is observation. So scientists first have to observe things that happen in nature, and then off of those observations, they will ask questions. Based on those questions that a researcher is interested in answering, the researcher will suggest a hypothesis, and this is to explain our observation or to explain that question. It's basically you're trying to answer that question that you're interested in based on your current knowledge. Based off of that hypothesis, we will generate a prediction. Predictions are very specific and they can be tested. So the next step is to design a test of that prediction or those hypotheses. There are different types of tests. You can test things by gathering more data, or you can test things by designing and running an experiment. We're going to focus on this experimental design part. When you collect that data, you can analyze the results, you can graph the results, and use statistics. Based on those results, we can then go back to the beginning and we either are going to accept our hypothesis, so our data matches what we thought was going to happen, that's accepting. We can reject our hypothesis, our data does not match our hypothesis or prediction. And oftentimes when a hypothesis or prediction is rejected, it will be modified, and you will continue to go around in a circle using the scientific method. Right in the middle, we have communication. Communication is a big part of the scientific method. Scientists will communicate with other scientists during any part of the scientific method. In addition, scientists will communicate their results by publishing it in a scientific journal. So we're going to go through these steps. So an observation, it's just a description, measurement, or record of any object or phenomenon. So in the case study in your book, we have scientists that are researching bats, and they're specifically looking at what is causing bat colonies to die. So the scientists can go in to the caves where the bats live and they can make observations. Based on those observations, we're going to create a hypothesis. And a hypothesis is a very informed, logical, and plausible explanation based on your current knowledge of how the world works. And it explains those observations that scientists are looking at. This is stated as a statement and not in the form of a question. This hypothesis we then use to make predictions. Predictions are very specific, so they are testable. We can design an experiment to collect data off of these predictions. They are falsifiable or refutable. This is the part where we can reject the predictions. Predictions can also be supported. And we say that we can support a prediction or support a hypothesis. We can never prove anything. Because there can always be an experiment that is done that will falsify a prediction. But we can never prove anything because we can't collect all the data that's possible in the world. Another thing about predictions is they're very specific or precise. And they're given in an if-then statement. And I'll give you an example of this when we get to our example. So to go along with these hypotheses and predictions, experiments can support a hypothesis, but we can never prove anything. So we can't know anything for sure. So media often use the wrong terminology, and that's done to get your attention. But when media claim that something is clinically proven, they should not be saying that. They should be saying that something is clinically supported. But proven gets more attention in the media, so things like newspapers, on TV, any advertisements will use proven. And this will lead into chapter 2, so looking at how our scientific literacy can help us really decide how we should look at different scientific claims. Off of our prediction, we can create an experiment. So an experiment, it is a repeatable manipulation of one or more aspects of the natural world. And repeatable, that could be within the experiment. This also means that the experiment itself can be repeated by other scientists. So to make a good experiment, there are a few terms that we need to know. One of them is the independent variable. So an independent variable is a variable that the scientist is researching the variable that they are manipulating. The variable that is being collected or measured is called your dependent variable. So this is any variable that responds to or could potentially respond to those changes in your independent variable. Another way to look at these is your independent variable sometimes is called your experimental variable. And the dependent variable is oftentimes referred to as the measured variable. So there's another way to look at them. A third way to identify these variables is to look at the prediction. So in a prediction, when it is written, it has that if-then format. So the variable that follows the if is your independent variable. So this is the thing that you as a researcher are changing. It's what you're experimenting on. What follows the then part of your prediction is the dependent variable. This is a variable that you are going to measure in your experiment after you apply your independent variable. In addition to having your two different variables and identifying those, experiments are often divided into two groups. One group is called the control group. This is a group of test subjects that is maintained under a standard set of conditions, and no change is made in that independent variable. So they will get what they have always gotten in the past. You're not changing anything in this control group. The other group is your experimental or treatment group. So treatment group or experimental group, this is where you're going to maintain them under the same set of conditions as a control group. So everything is the same between your control and treatment group, except for your independent variable. That is what you are going to manipulate. So you really only want to change one thing. Everything else will be controlled. So to give an example of this, or to pull this scientific method together with these different terms that we just talked about, we're going to look at a drug example. So pretend you work at a pharmaceutical company and you observe that a drug, we'll call it drug X, it lowers blood pressure in mice. So that is our observation. So based on that observation, we create a hypothesis, and we are going to look at if drug X would be a safe and effective treatment for high blood pressure in humans. And we can go from mice to humans because we know that mice and humans are very similar to each other physiologically. They're both mammals, and a lot of drugs tested on mice have shown the same effects on humans. So our hypothesis is a very educated guess about what we are interested in. Off of that hypothesis, we will create a prediction. And again, that is written in the if-then statement. So if drug X is a safe and effective treatment for high blood pressure in humans, so here our drug X is following that if part. So that is going to be our independent or experimental variable. That is the thing that we are looking at, that we are interested in, the thing that we're going to change in our experiment. Then 10 milligrams a day of drug X will lower blood pressure in people that have high blood pressure after 1 month of taking the drug. The then part, this is where we're going to find our dependent or measured variable. Okay, so that's going to be the blood pressure, what we're actually going to be measuring after individuals take the drug. So we can take that prediction and we're going to set up our experiment with our control and treatment groups, and then we'll have our independent and dependent variables. So we are going to collect a bunch of test subjects, in this case humans, that have high blood pressure, and we are going to split them into 2 groups. Those 2 groups, one of the groups, group 1, is going to receive our drug X over here. So this group is our treatment group. Because we are giving them the drug, we're changing that one thing about their lifestyle. Everything else, we don't want them to change. We don't want them to change their physical activity. We don't want them to change their diet, etc. Group 2, this is going to be our control group. So they are going to maintain their diet, their lifestyle, etc. We're also going to give them a placebo, which is a sugar pill, so that both groups are taking a pill. They don't know which one they're taking. This is referred to as a blind study. We as the researchers, we know what group is getting the drug and which group is not getting the drug, but they don't know. So this is to control for any biases that may occur with taking a pill. So we can control for that as well. But we know they're just getting that sugar pill. So our independent or experimental variable is whether they're getting that drug or not. So the type of pill that they are taking. So we'll give them 10 milligrams of that pill every day for 30 days. And after those 30 days, those people are going to come back in to the clinic, and we're going to measure their blood pressure to see if it stayed the same or if it went down. So that is our measured dependent variable, that blood pressure. So we have our 4 things identified. We have our treatment group, we have our control group. We apply our independent experimental variable to those groups. So one gets the drug, one gets a placebo. After they take it, as specified in the prediction, we will measure our dependent variable, which is blood pressure in this example. We collect that data, and then based on that data, either our hypothesis will be supported, so that drug lowered blood pressure in our treatment group, so that supported our hypothesis, or the drug did not work. It did not lower blood pressure. That is leading to the no, and we can modify and reject our hypothesis and come up with a new experiment. So other experiments that they could do is they could increase the amount of drug that the treatment group is taking. They could have that treatment group take the drug longer. They could look at other factors that may play a role in that drug. Maybe they need to take it at the same time every day, and they didn't control for that the first time around. So you can always modify those specific parts of the prediction and come up with a new experiment. So these experiments that are done, they will lead to scientific facts and theories. So a scientific fact is a repeatable observation of the natural world, and these facts can change as observations become more accurate. A scientific theory, this is a hypothesis or a group of related hypotheses that have been repeatedly confirmed by diverse independent researchers. And we're going to be looking at a couple of different theories in this course. We'll look at the cell theory first. There is the theory of chromosome inheritance. There is the theory of evolution. And those are just a couple that we'll see as we work through this semester. Once research has been done or an experiment has been completed, those researchers will write up that experiment and they will publish it to a scientific journal. And scientific journals are going to publish this research only after it has been judged by experts in the same field as the research. And this is referred to as primary literature, and we'll talk more about this in Chapter 2. So this judging by experts in the same field is called peer review. So these peer-reviewed publications, they prevent any types of mistakes in experimental design. So for example, some experiments, maybe they forgot their control group, or their two groups were not treated the same, like there was a difference in diet, etc. We also prevent misinterpretation of the data and fraud. And this is really important to understanding scientific literacy, so making sure that what you are reading is peer-reviewed really helps to make us make good decisions about different types of science and what is being presented to us in the media. So biology is the science of life or study of life. We took a look at that science part, and we'll come back to that in Chapter 2, talking about science literacy, how we can evaluate scientific claims, and the criteria for that. The next part is looking at life. So living things are organized into a classification system, and one of them is the biological hierarchy. So all living things are composed of atoms. So we're starting right here. Those atoms come together to form molecules. Those molecules will create cells. Cells are the smallest unit of life. So they will show all of the characteristics of life that we will get to next. Cells come together to form tissues. Tissues create organs like skin, stomach, brain, etc. Those organs create organ systems. In humans, we have 11 different organ systems. So our respiratory system, our reproductive system, our digestive system, etc. Those organ systems create one individual organism. If you have a bunch of organisms of the same species, that creates a population. If you have populations of multiple species, that is a community. And communities contain all the living things in an area. Those living things then interact with any non-living things like water, rocks, etc. And that creates an ecosystem. So ecosystems are living and non-living things. Those ecosystems can then be found in different biomes. Biome examples include desert, freshwater, prairies, deciduous forest. And all those different biomes come together to create our biosphere, which is the earth, including any land and water, plus our atmosphere, so the air that surrounds the earth, where you can find any living things. So these living things, they have certain characteristics to them. So if we look at them, there are 6 characteristics of living things, and this last one is just saying if it's living or not. So if you look at a rock, rocks are not living, so they do not have the characteristics of life. Viruses are interesting. They do have some characteristics of life, but not all of them. So technically we say they are not living, although some people will argue that they are living. And then we have our fungi, plants, animals, protists, bacteria, archaea. they show all 6 characteristics of living organisms. So these 6 characteristics, we'll go through. The first one is that all living things are composed of one or more cells. And there are 2 different types of cells. There are prokaryotic cells. These include the bacteria and the archaea. They are very small and simple. The second type of cell are the eukaryotic cells. Eukaryotic cells, we find these in animals, plants, fungi, and protists. They are larger and much more complex. And you can see this based off the pictures. These eukaryotic cells, there are a lot more structures inside of the cells compared to our prokaryotic cell. And those structures in the eukaryotic cell are called organelles, and we'll get to those organelles and their structure and function. The next characteristic is that living things reproduce autonomously using DNA. And living things will either reproduce asexually or sexually. So they will use cell division techniques, either mitosis or meiosis. The next characteristic is living things obtain energy from their environment to support their metabolism. So metabolism is all the chemical reactions in a cell. In our metabolism, we need energy and nutrients, or food. So all of this energy comes from the sun and flows into our energy cycle in our biosphere. And this is an image from a different book, but I really like it. It summarizes this energy cycle really well. All the sunlight coming in from the sun gets absorbed by plants and other organisms that can do photosynthesis. Those organisms, they take carbon dioxide and water along with that sunlight. They run it through photosynthesis to create organic molecules, and this will be the food, and oxygen. That food and oxygen is then taken in by cells, and it's used in cellular respiration. Cellular respiration is where we take organic food molecules and turn that organic food into usable energy, called energy intermediates. One of the energy intermediate molecules that we use the most is ATP, or adenosine triphosphate. The next characteristic is that living things sense the environment and can respond to it. And we are very familiar with this because as animals, we can move around in our environment. Other organisms can also move, like bacteria. Plants, which we usually don't think about as responding to their environment, they can respond, but they do it on a much slower scale. So for example, plants, if you place them next to a window, they will bend towards the light. Another example, we have a hare that will change fur color depending on seasons to help it camouflage into its environment to avoid predators. Living things also maintain a constant internal environment. This is called homeostasis. An example of this is temperature. So we are endotherms, like the bird shown up here. These are also referred to as warm-blooded. Endotherms are able to maintain their internal environment by their nervous system So for example, when we get cold, we start to shiver. We don't think about it, it just starts to happen. When we get too hot, we start to sweat to cool ourselves down. So we are maintaining that body temperature. Other organisms are called exotherms or cold-blooded. Instead of having those normal reactions like shivering or sweating, exotherms will move in their environment to change their temperature. So this is where if you have a reptile, in order to increase their body temperature, they will sun themselves on a rock. When they get too hot, they will go under the rock into the shade. So they're still maintaining their temperature, they're just doing it in a different way. They're still maintaining that homeostasis. The last characteristic of life is that living things can evolve as groups, and specifically populations will evolve. And this is what leads to the big diversity that we see in all living things. So all living things, whether if it's a bacteria, an elephant, a fish, a Christmas tree, so a plant, they all have these common characteristics, but we also have this big diversity. And that diversity comes from this evolution, or being able to adapt to different environments. So in this chapter, we looked at biology. We first looked at that science part and how that occurs using the scientific method. And then we went to the life part and what characteristics those living things will have.